In the mid-1860s, Swiss chemist Johann Fried Miescher stole some bandages from a trash can. Using these bandages for his experiment, Miescher noticed a substance he was unable to identify. Little did Miescher know he was actually peering down at a DNA molecule. As time went on, more and more scientists researched this mysterious molecule. However, the most decorated breakthrough is the introduction of the double helical model of DNA by Watson and Crick. These individuals accepted Nobel Peace Prizes for their works despite the help that they had received from Rosalind Franklin, because she was dead. Watson and Crick's DNA model is still used to this day. However, upon closer inspection, more detail about this molecule becomes transparent. To begin with, some basic building blocks of DNA are its nucleic bases. These bases include adenosine, which binds to thymine, and guanine, which binds to cytosine. Adenosine and guanine are recognized as purines, appearing larger in structure. Cytosine and thymine are recognized as pyrimidines, appearing smaller in structure. Each complementary pair of bases is joined by a hydrogen bond. This helps the DNA maintain its double helical structure. The outside end of every base will bind to a deoxyribose sugar, which will, in turn, bind to a phosphate group. This hierarchy of phosphate groups, bonding with sugars, embodies what is known as the DNA backbone, which acts to add support to the complementary nucleotide of DNA. In its totality, DNA functions as the encyclopedia for cellular function, providing directions to other polymers, namely proteins. Now, on occasion, cells containing DNA will need to divide. When this happens, we can't have DNA absent in the daughter cells. To that end, DNA will replicate itself. In replication, there are 4 primary enzymes: helicase, primase, DNA polymerase, and DNA ligase. To begin with, helicase unzips your DNA at points of origin. Primase follows, carrying a primer which acts as an indicator for the DNA polymerase. Then, the DNA polymerase will build and attach sections of new DNA to the parent strands. Finally, DNA ligase appears to act as an adhesive, holding the new sections of DNA together. DNA strands are identified by their directionality. That is to say, they're broken down into 2 categories: strands running 5' to 3', and strands running 3' to 5'. Between the 2 strands, one will run 3' to 5', known as the leading strand, and the other will run 5' to 3', known as the lagging strand. Respectively, these strands' complementary strands will run 5' to 3' and 3' to 5'. As previously mentioned, DNA is the cell's encyclopedia. DNA has a counterpart, RNA, which exists as a single-stranded replication of a section of DNA. RNA is used as a message to shoehorn the synthesis of proteins. However, it's the process that creates this protein that proves the most interesting. To begin with, the protein creation process is broken up into 2 sections: transcription, which serves to synthesize RNA within the nucleus, and translation, which serves to synthesize proteins within a ribosome. To digress, transcription begins with an RNA polymerase binding to a promoter. This RNA polymerase reaches a terminator. The RNA transcript is released, which acts as a copy of DNA. Then, through RNA processing, the RNA transcript is converted to mRNA. Keep in mind that RNA, like DNA, contains the nitrogenous bases A, meaning adenosine, G, meaning guanine, and C, meaning cytosine. However, unlike DNA, RNA contains the nitrogenous base U, meaning uracil. Uracil acts as a substitute for DNA's thymine. At this point, the mRNA leaves the nucleus and travels into the cytoplasm toward a ribosome. This signals the beginning of translation. From here on out, the mRNA's bases will be associated together in pairs of 3, being recognized as codons. Upon arriving at the ribosome, the mRNA is approached by tRNA. The tRNA contains an anticodon, which acts as a counter to the mRNA's codon. The tRNA searches for a codon on the mRNA containing its complementary bases. Once located, the codon will bind to the tRNA's anticodon. Additionally, the tRNA will attach its amino acid to the mRNA. As more and more amino acids bind to the mRNA, they compile to create protein, a polymer of amino acid monomers. The protein can then be used for cellular processes.